Thank you so much. JT, start that timer. Keep me honest. Okay, really excited to be here to talk about what we've been doing, especially with base editing screens. It's not the only way of perturbing the genome by any means, and we love doing this with all sorts of perturbations. So the idea of doing large-scale pooled screens where we start by synthesizing oligos and we introduce these things into cells via lentivirus. We select the cells with some sort of positive or negative selection pressure, and then at the end of the day, you read it out, usually via Illumina sequencing, although there's many new and exciting other ways of reading out the effects of perturbations that we've heard about and will hear about later today. Here at the Broad in the genetic perturbation platform, we tackle this in a very collaborative manner where the things that scale really well, that platformize really well, we do in blue. And we rely on our collaborators who are experts in their particular cell model or mouse model or whatever the case may be to do the parts that are highlighted in red. And this scales quite well. In the year 2023, we made an awful lot of custom libraries, a lot of lentivirus, don't tell Cambridge Health and Safety. We collaborated with well over 100 PIs, not just in the Boston community, but really from around the world. Here I'm going to talk about base editors. So base editors, as we've already heard about, this is a way of doing chemistry on individual bases driven by a Cas protein. I even took out the introductory slides because we already know this. So we wanted to understand how could we use base editors to study protein small molecule interactions. And for that, we wanted to start with a positive control, basically. In this case, the positive control is the inhibitor venetoclax and its target BCL2. So venetoclax was developed to inhibit BCL2. BCL2 is an anti-apoptotic gene that cancers often upregulate. So the hope is that if you inhibit the anti-apoptotic gene, now the cancer cells will die. And indeed, this was approved in 2016 to treat a variety of leukemias. And it was approved, so it does actually work. But of course, resistance inevitably arises. And that's highlighted in the crystal structure shown on the right. So this is a clinical study of people who relapsed on venetoclax treatment. And you can see in orange is the venetoclax small molecule, and highlighted in blue and purple are residues that they found mutated in patients that could no longer now respond to venetoclax. Now, obviously, these are not loss-of-function mutations of BCL2, because it still has its anti-apoptotic function, but it can no longer bind to the small molecule. So we asked, could we have discovered this using a base editor screen? So we took a leukemia line, in this case MOLM13, we designed all possible guide RNAs that we could to introduce base edits, either using A to G or C to T base editors, and we introduced it so we now have a pool of mutations, of point mutations in this particular line. Now, for those who have done large-scale CRISPR screens, normally you're working with tens of thousands of perturbations and thus tens of millions of cells. Here, this is tiny. This was like 1 10-centimeter dish. So very easy to set up and screen. And then at the end of the day, what we're looking for is what are the mutations that enriched in the presence of venetoclax. And you do that, and so each dot here represents a guide RNA with either of the two editing types along the length of BCL2. And there are several dots that gave rise to resistance, and when you map those onto the crystal structure of venetoclax bound to BCL2, you see they are also right in the binding pocket. Now, this is a case where we already knew what answer we should get, but we got it. So we felt good that this is a technology that can be used. If we didn't know at all where is venetoclax binding to BCL2 or is venetoclax binding to BCL2, this would be very strong evidence that it indeed is. And when you go and validate a particular hit, for example, aspartic acid 103 is normally in the binding pocket, and it makes some nice hydrogen bond contacts with venetoclax, but the base editor changes this to a glycine. You now lose that hydrogen bond, and that explains why it's no longer binding. Now, I want to be clear, and I think this is something that we really need to be careful of when using these editing technologies, is that validation is really important. It's important in any sort of screen that you do, but with CRISPR knockout screens or activation or interference, you get a lot from the redundancy of your library. You can have multiple guide RNAs of different sequence that trigger a knockout or whatnot. And so you can have lots and lots of guides that all vote together and say like, yep, this really is involved in the phenotype. But with base editing screens or prime editing screens, it's generally only one guide that causes your phenotype. And you really can't say with certainty from a primary screen, well, what edit actually happened at the locus of interest? So to do that, one pretty straightforward validation approach is you now take your one guide, you introduce it into your cells, and then you apply the same selective pressure. So it's the exact same assay that you did in the primary screen, but then you sequence the alleles that come out at the end, and you see what actual alleles are enriching in the context of the selective pressure. So this is an example, not with BCL2, but with another anti-apoptotic gene, MCL1, and its inhibitor. And you can see you get, and this was using a C to T editor, you get all sorts of different alleles. And so you sequence them and you ask the question, well, what alleles enriched, what alleles depleted? And then you can translate this back into amino acid space. And you can see that, okay, well, there were several alleles, all of which had a valine at position 227. Those are all the ones that enriched. You can see that methionine, that just comes along for the ride. You can also see some alleles that failed to enrich, including one that has the valine. But in that one, there was also a nonsense mutation 2 codons down. So in a very nice way, you can get, some might look at this and be like, oh, this is kind of a pain because you're getting all this allelic variation. And yes, it is. It would be great if we knew exactly what every guide RNA was going to do. But I still think it's very pleasing to look at this and say, like, okay, well, what alleles really enriched and which ones did not? But I think this is a really critical validation step for any of these sorts of screens that we're doing, not just to rely on here's what we think the guide RNA is doing, but to actually validate what's happening in the cells. And indeed, in this particular case, it's also right in the binding pocket for where this small molecule binds. You can do this not just with one small molecule at a time, but this scales particularly well. Here, we did a screen where we tried 5 different PARP inhibitors. PARP inhibitors are clinically approved. They synergize very nicely with tumor types that have lost BRCA genes. And you can see in some cases, there are the same point mutation gives rise to resistance to all PARP inhibitors. But here's an example where for 4 of the PARP inhibitors, this is a guide RNA that provided resistance to 4 of them, but actually the opposite phenotype for 1 of the PARP inhibitors. So you can really, again, if you had no idea what what PARP looked like or how it was complex with the different inhibitors, you can use this to start to understand, well, what's important for each small molecule that I've developed, which again, these have already achieved clinical efficacy and approval. But if you have a panel of small molecules against a target that you're starting to work on, I think doing screens like this would be really valuable to understand what are the similarities and differences across my panel of small molecules that I'm considering moving forward. Here's a very recent example of now moving these base editor screens sort of further up in the pipeline. So this is an example from Scott Armstrong's group where they were looking at this inhibitor, revumenib. I don't know if I'm pronouncing that right, but that's how I pronounce it. And they were doing these base editor screens at the same time as they were doing the clinical trial. And they concluded that consistent with the genetic data in patients, these particular mutations that they found using these base editor screens worked in both their xenograft model and in what they were seeing in patients. And what you can also see from this, so they found 2 mutations that are in red and maroon in the graph on the left that clearly provide resistance, but for a different menin inhibitor that is being developed, this peptide-based inhibition on the right, you don't see nearly as much resistance from those particular mutations. So in some glorious future, we'll be able to understand, well, here are the different inhibitors one might want to use based on any particular resistance mutation that can arise. So here's an example of what you don't want to happen and why doing these screens is critical. So reclinostat, this was first described in the year 2012. It had a different name back then. And it was originally developed by this company called Acetylon, which was then purchased by Celgene, which was then purchased by BMS. I guess that's a pretty typical lifespan for these things. And in 2023, there was a paper that appeared on the network-based assessment of HDAC6 activity predicts clinical responses to this HDAC6 inhibitor reclinostat in breast cancer. So, and they say in the abstract for this, that they had designed a phase 1b dose escalation trial to evaluate this. So this is now going into people who have received a diagnosis and they're now being treated with this. So Jason Scheltzer, who was at Cold Spring Harbor, is now at Yale. So this was, how do I go back? So note that this paper was 2023. And then in January of 2023, Jason sent this paper in saying that reclinostat is not a highly selective HDAC6 inhibitor. And he goes through the literature on the development of this small molecule, and he makes two very salient points. First, 3 independent labs have engineered genetic knockouts of HDAC6, and whether you have HDAC6 in your cells or not, this small molecule is effective. If you look at the Broad DepMap project, there are 0 out of 1,000 cell lines screened with CRISPR and 0 out of 710 screened with RNAi that seem to care about the presence of HDAC6. And to this, I would add, at no point did anyone find a resistance-conferring point mutation. So I would argue, as we're thinking about moving small molecules forward and proving what the target of a thing is, that is not a trivial experiment to do. But if you can find a point mutation that gives you resistance to your small molecule, that's very good evidence that it is the target that you think it is. Okay. So everything I've talked about so far is using positive selection screens where even low efficiency can lead to enrichment of a variant of interest. But a lot of times we want to do negative selection screens. We want to see, well, what is a loss of function mutation, and can I see that? So to see where we are with that, we looked at the gene P53. The reason we picked this one is obviously it's extraordinarily well studied. And not long ago, Andrew Giacomelli and Bill Hahn's group did a very similar screen, the exact same screen, using an open reading frame-based library to look at variants in P53. So what's nice about P53 is that if you treat cells with Nutlin, then in this particular cell line, A549, loss of function mutations are known to enrich. Whereas if you treat with etoposide, a DNA damaging agent, then you actually need P53 to initiate the repair, so loss of function mutations deplete. So you get both sides of the screen that you want to do, or both sides of the selective pressure. So this is what the data look like using either ABE and CBE, or treating with Nutlin shown in red, or treating with etoposide shown in blue, again across the length of P53, and you see that there's a very nice mirror image. The red dots generally go up, and the blue dots generally go down. And then when you look at what domains of the protein you tend to get most of the action in, and you compare it to where, I believe this is from Cosmic, where are mutations found in cancer patients, you would learn, if you had no idea that P53 was binding DNA and doing cool things, you would have learned it from doing this sort of screen. I think what's particularly nice about this is I think with the base editors as they are now, they are efficient enough to allow people to do this experiment in a pooled setting. So this is a paper now at this point, maybe almost 10 years old, from Junwei Shi when he was a postdoc in Chris Vackage's group. Junwei is now at Penn, where they were using, in this case, CRISPR-based knockouts, because base editing had not yet been invented, to tile across the length of, in this case, the gene BRD4, but they did this for a number of other genes. And in the assay that they were using, they were able to say, like, well, when we enrich for a domain is important if in-frame indels are also detrimental. So what they're looking at here is can you in an unbiased way map a protein and figure out what domains are particularly important. However, this was done in an array-based, so one perturbation at a time, read out in 96-well format, in order to achieve the resolution necessary to build a nice map. This is not an approach that scales to pooled screening. You just don't have the resolution of being able to do a very nice ratio like this. But I now think we're at the point with base editors that they are efficient enough that we can now do this. So if we take our tiling of P53 and we ask, just based on where the domains are annotated, we got no hits that were in between the domains. But over 30% of guide RNAs that were in the DNA binding domain led to a meaningful phenotype. So I think that if we have, now again, P53 we know a lot about, but for proteins that we don't know and we want to understand, well, where is the action, which domain actually matters for this protein of interest, I think these unbiased tiling base editing screens are a way to get us there. Now, if you were squinting very closely, you may have noticed that, well, red tended to go up and blue tended to go down. There were a couple examples where that wasn't true, including right at the end terminus of the protein. So this is the exact same data, but now plotted in a different way, where we're comparing the screen result either with Nutlin on the x-axis and etoposide on the y-axis, and you see that anti-correlation or the mirroring, but you also see a small number of dots that now move completely off the diagonal. We were interested in what those were doing. So we validated them. So we put these guide RNAs into cells and sequenced what was actually happening. And for these two guide RNAs that we validated, we saw that what was happening was we were actually mutating the methionine start codon to either a threonine or an isoleucine. So we were losing the normal place where translation starts. And we could see that via Western blot as well. We see a short isoform of p53 increase in abundance when we've introduced this mutation. And one of the benefits of doing anything with p53 is that it's been studied to death. And it turns out there is already a well understood or reasonably well understood secondary site for translation, methionine 40. It's not clear to me from reading the literature if this is due to scanning past the first one or due to a different splice isoform. I found papers saying either. But on some level, I don't care. What you can see, though, is that if you lose the first 40 amino acids, you lose a whole chunk of the first transactivation domain. So you could certainly, it would make sense that now the transcriptional targets being regulated by p53 are in fact different, leading to these differential outcomes. And indeed, there's this delta 40 mutation has been observed many times. This is not something that we saw for the first time. And of course, if you're doing these sorts of screens, you don't need to look only at coding regions. So this is something that Ryan in the group has done pretty recently, where we wanted to understand the 3' UTR of CD274, more famously known as PDL1. So we again did a C to T or an A to G base editor screen, but now we're tiling just the 3' UTR. CD274 is on the surface, so you can do flow cytometry, select the ones that are high expressors, low expressors, and build a nice map of where are the functional elements in the 3' UTR. So we do that, and we can find some regions that lead to increased expression, decreased expression, et cetera. We can go to validate this. Now, of course, these are small effect sizes, right? So here's one particular guide RNA that scored very strongly in the screen, but it's not causing a complete loss of PDL1 levels, but it's shifting the expression just a little bit. And we can then isolate those cells, do the endogenous sequencing as we did before, and you can see, as we have more mutations, so if we mutate all three T's to C's, or this is on the bottom strand, so it was an A to G editor, which is why it's T to C, not C to T. If you mutate all three, then you see a bigger change in expression than if you mutate one or two. We then very recently asked, well, would we have gotten this via a luciferase assay? So we took that 3', we took a portion of the 3' UTR, not the whole thing yet, but we took a portion of the 3' UTR, and we asked, and so what we're looking at is a standard luciferase assay. Some positive controls are if we put in a binding site for either a microRNA or for an ARE, which is a destabilizing element. Those are the two shown on the right. We see a nice reduction. And then here's what we see for the different alleles that we put in. But then when we compare what happened in the base editing when we sequence the alleles versus what we're seeing with the luciferase assay, we're not really seeing the same thing. Now, I would argue that it's hard to argue that the sequencing of the alleles that came out of actually sorting the cells into PDL1 high and low, I think this is the right answer. And the luciferase is leading to an artificially wrong call, especially for that dot shown in brown where all three nucleotides are mutated. And of course, luciferase assays are great. My entire graduate career was like basically doing luciferase assays. So I believe that they can work, but you always need to validate that the observation you're seeing is true at the endogenous locus because these are artificial systems. Oops. Let's see. So those are screens where we're looking at one gene, or in this case, one UTR at a time. But one can also use base editing to screen lots of different places at a time. So this is actually one of the first base editing screens we did. We took everything that we could find in ClinVar that was at the time editable by a C to T editor, because A to G editors were still being worked on. And you can, design, in this case, 68,526 guides that we thought would introduce a variant that was found in ClinVar. We did a couple screens with this in two different cell lines where we either subjected the cells to cisplatin to trigger DNA damage or hygromycin to trigger translational stress, and then we sequenced the guide RNAs. In this case, we saw that there were a small number that scored really strongly, and the ones that were, if we just classified by is the gene involved, annotated, involved in DNA damage, that represented a tiny fraction of the library, but it actually represented a huge fraction of the hits that scored in the presence of cisplatin, so we felt pretty good about that. And if you look at some of the genes, they are the genes that you would expect. A lot of these were categorized as variants of uncertain significance at the time. But I want to be very clear, we did not validate all of these guides. So I would not say, well, this guide RNA, we think it introduced this variant, and we think we should reclassify it. I think the strength of evidence of a primary screen like this is not sufficient to do that. I want to be very clear. But it certainly gives you places to look for hypotheses to follow up. One screen we did that was particularly nice is we also did a counter screen where we took the same library but now paired it with wild-type Cas9, so it's acting as a nuclease and presumably the vast majority of the time introducing loss-of-function mutations. And we can then compare those two. So for example, the dots, the guide RNAs in the bottom left, those are the ones that showed a loss-of-function phenotype with both the base editor and the nuclease. But there's a small number of guides that only showed a phenotype with the base editor, but not with the wild type. And I think this is a really cool way to identify rare dominant negative alleles. And here's one example. So this is a guide RNA that introduces a G244S allele in this gene TUB4A. TUB is a tubulin gene. And so the way this seems to function so this is a gene that is known to give rise to leukodystrophy, lipodystrophy, I think. If you have this mutation, so a point mutation, then this broken version of tubulin will go into the growing tubulin fiber and prevent it from being extended. However, if you lose it completely, then TUB4B takes over. So it's a way of finding, again, in this case, a well-understood dominant negative allele, but just doing this comparison of point mutants that show very different effects with the knockout versus the point mutant is a way, I think, to identify these in a more unbiased way. And I think there's a lot you can learn from dominant negative alleles. They're rare, and so finding them in patients is really, really hard. But I think this is a way that one could screen for them and find more of them in an unbiased way. There have been a couple of other screens that have been taking the same idea. So, we screened lots of things that were found in ClinVar. This is work from Ben Kravat's group where they did a screen of all the cysteines that they could perturb. Cysteines are a very exciting target for the chemists because there's now ways you can covalently attach things to cysteines. So they wanted to do assigning functionality to like all cysteines in the genome via this base editing approach. Here's a paper from Sam Myers, who was previously a postdoc here at the Broad. He now runs his own group out in San Diego. Same idea, generally speaking, but he wanted to look at all potential phosphosites. So he started by doing some proteomics to identify what are the relevant phosphosites for my pathway of interest, and then he did a base editing screen to change as many as he could to understand which ones were really important for signaling. So where are we with base editing screens? We think the C to T and the A to G editors enable mutation of endogenous elements, the expanded PAM-Cas proteins that I think I saw Russell in here. Russell, where are you? Yeah, the expanded PAM variants that Russell has developed have been extraordinarily helpful for increasing the density of these screens. Finding drug resistance mutations in a number of genes. You can identify loss of function mutation and endogenous regulatory elements, so all great stuff. I think a real use case for these is confirming the on-target activity of less characterized small molecules, mapping the functional landscape of an uncharacterized gene. I think these are really the sweet spots for base editing. I just want to compare it to, there's lots of ways of doing this, and I'd certainly be welcome for anyone to correct any of the boxes in this chart. I think this would be a great debate to have. For the base editing screens, where they're not as good as other approaches is it's not saturating, right? The base, as I showed in the alleles of MCL1, you get lots of different edits and you got to figure out which one is actually driving the phenotype because you're mutating multiple Cs or As in a particular editing window. And obviously, if that's all you can do, then you can't change all possible residues. But they have the benefit of being extraordinarily scalable. It's pretty easy to do 100,000 guide RNAs this way. They're really cheap relative to these other approaches because you're really just sequencing the guide. They have the benefit of editing the endogenous locus. You're not relying on a reporter. So I think they have a really useful place in the toolkit. So what I would suggest, or a potential screening campaign, start with a tiling base editor screen. They're small scale, it's easy to do, they're very low risk, and you can find regions of interest in your protein that you're interested in or your regulatory element. Then follow up with either an open reading frame library or maybe a prime editing screen. Those are starting to maybe have enough signal to be generally useful. And then, of course, if you really want to know what's going on, then introducing the exact mutation at the endogenous locus with HDR or prime editing is the way to really nail it. So, when in doubt, screen it out. In every technology, every perturbational technology you throw at a model system is going to tell you something new and different. In terms of where we are with actually doing this stuff, so delivery is still a challenge for lots of different model systems. I think the work from Alex Marson in T cells shows that if you work at it hard enough, you can really wrestle it to the ground. But of course, there's a lot of model systems out there. I'd say our ability to control CRISPR technology spatially and temporally, it's possible, but that doesn't mean it's easy. And that's another thing that often needs to be wrestled to the ground in your particular model system. And of course, the path from a gene that scores as a hit or a variant that scores as a hit to mechanistic understanding is still not trivial. Higher dimensional readouts, whether it's single-cell RNA sequencing or pooled optical screens, that's more data, but that doesn't necessarily provide more insight. It can, but it can also be difficult to wade through, so it's not a silver bullet. And to absolutely emphasize something that Heidi mentioned repeatedly in a very important way, accurate models of human disease, we really need those. You can do these screens till the cows come home, but if your model system isn't relevant, then you're not actually learning anything that moves the needle. I want to give a quick shout out to Sayeh. I know Jordan in her group presented yesterday. The G2P portal developed here at Broad is really helpful for taking the results of base editor screens, integrating lots of information from other places, not needing to go to 5 different websites yourself, and visualizing your screen results. Working with her and others on this has been really, really helpful. And so I want to thank everyone in GPP. I highlighted the folks in the lab who were doing the work. But I especially want to highlight Priyanka and Laura and Feng Yi, computationalists in the group, who help us make sense of all these data, and especially Allison and Amy, who also help us to coordinate a lot of our work. Along with all the other teams in GPP, our production team that makes it easy for us to collaborate with hundreds of labs, our informatics team that creates tools that people love to use, our operations teams, our screening team for the whole DepMap project, and of course the senior director of the group, Dave Root. And with that, I'm happy to take any questions.
Thank you, John. That was absolutely fantastic. What a tour de force. We've got time for a few questions. If you'd like to make your way to the microphone or put your hand up, please say your name and where you're from. Alan.
Alan Rubin, Walter and Eliza Hall Institute. You've obviously had a huge amount of success using z-scores to calculate the functional effects, but I think we know from demutational scanning that they're not really normal. They're tend to be strongly bimodal. So I was wondering if you could comment on, like, was this just sort of the best of the imperfect models that you tried? And also, is this like an area that you're working on to maybe do some more modeling of the scores, especially to kind of recover some of these smaller effect sizes?
Yeah, so I think there are two answers to that, and I'll give you the professional answer and then my real answer. The professional answer is yes, we absolutely need to figure this out, and in a more statistically rigorous way, identify to put some meaning on that x-axis for like what is the effect size of this variant. My glib answer is, for the purposes of what I'm doing, I don't care. I just want the top hits, and no amount of statistics is going to change what like the top hit was. And since I think in the discovery phase, you always need to do the next experiment, you're gonna run out of energy to keep validating stuff before you worry about the statistical certainty to it. So it's an extraordinarily important problem, but I think at this level of discovery, it's not a critical one to solve before you can move forward with the results from your screen.
Doug?
Doug, hello.
Hey, hi. Nice to see you. I really enjoyed the talk. It was great. We have got a little further than 10 exons in one student, but
Yeah, I should update that slide. It's a joke I like to make for Greg.
Here who have worked hard, so I want to say that. But I also just wanted to sort of ask maybe the what's next question from Alan, because I do think it's really important. Like, do you think we'll get to a place where we can move base editing from a screen to like a score for every variant, where we actually can just be done with classical mutational scanning because we can score every variant? Or is that not the direction things are going to go?
I don't think that's the direction in the next couple of years. It doesn't seem to me likely that we'll, there are approaches out there to try to take a guide RNA and predict what the edits are going to look like based on sequence features and other things. Those are good, but they're not perfect by any means, and they also seem to vary as you go from one cell type to the next to some degree. So as we're moving across model systems where you want to do the screen in the right model system, it'd be a ton of work to generate all those data. So I don't think a primary base editing screen, at least in the next couple of years, is going to provide enough specificity. Maybe with more and more generations of base editors or other similar-ish technologies, we will get there, but not anytime soon.
Okay, well, I will have a word with my colleagues back in Oxford about getting Pertable into the Oxford Dictionary.
No, please don't.
Thank you very much. That was fantastic. Okay, so we are...